Welcome back. Today we're looking at a really interesting piece on biodegradable aerogels, specifically the polysaccharide-based ones. It covers synthesis, biomedical uses, drug delivery, tissue engineering, that sort of thing.
Yeah, it's thorough, definitely shows a lot of expertise. But like we discussed, there are ways to perhaps elevate the connection between the bench work and the potential clinical impact.
Exactly. Let's dive right in with the first point. We felt it was about strengthening the connection between the new synthesis methods and actually tuning the function for specific applications. The document clearly details some cool production techniques, emulsion process, inkjet printing, that modified spray drying, which is great.
Methods, how you get a nice spherical shape or control the size really well.
Right?
And then separately, it talks about performance, like how the drug releases.
Yeah, and the link between the two, that direct cause and effect, where this process variable leads to this better clinical outcome, sometimes it feels a bit abstract.
Like the example with the surfactants, SPAN 80 versus SPAN 85.
Exactly that. In the alginate ketosand microparticles, it goes into detail on the process control, which is solid work, but it stops short, doesn't it? It doesn't immediately connect choosing SPAN 85, say, to really maximizing something crucial, like making it work way better for pulmonary delivery.
We see the pieces, the complex chemistry, the particles, but the why, the intentional engineering blueprint connecting them, isn't always front and center.
So it feels a bit like, okay, here's the tool and here's the problem it solved, but maybe not enough about why this specific design feature of the tool was the key.
Which leads to the suggestion it's really about weaving that narrative more proactively, telling the engineering story.
Yeah, make that link immediate, explicit. Show how mastering things like particle size or surface charge or tuning the porosity
Through these novel synthesis routes you're describing.
directly translates into solving a real therapeutic problem. Better bioavailability, better targeting, more stability, whatever it might be.
Okay, so let's make that concrete. We could use the pulmonary delivery example again.
Perfect.
The text mentions the fine control from inkjet printing, getting those highly monodisperse alginate beads, which is technically quite impressive.
Right. And that's the opportunity. Instead of just saying monodisperse, explicitly link that precision to hitting the specific aerodynamic diameter needed. What was it, 4.0 plus or minus 1.5 microns?
Yeah, the size that's required for optimal retention deep in the lungs, in the bronchi and bronchioles.
Exactly. Frame it like, we use inkjet printing because it gives us this precise control, which lets us hit the 4-micron target needed to stay in the bronchioles. That makes the synthesis choice a clear solution to a physiological barrier.
It shows intent. And what about the chemical side? The polyelectrolyte complexation, alginate, chitosan again? You noted the SPAN 85 samples gave a high positive zeta potential.
A positive surface charge. Yeah. Right now, it's presented more as a characterization result. We measured this.
Okay, so the suggestion is to push it further?
Definitely. Turn it into a functional decision. Show how that positive charge is being used. Is it an engineered strategy to make the particles stick to mucus in the lungs? Mucoadhesion?
Or maybe to target specific cells that have a negative charge?
Precisely. Then it's not just, we made this material and it has this property. It's, we manipulated the polymer arrangement in order to get this positive charge because that helps us achieve mucoadhesion or targeting. It demonstrates functional design.
That definitely shifts the focus from just describing the material to explaining the pharmaceutical engineering thinking behind it. It adds impact.
And thinking about that structure, the pores, the surface. It brings up that inherent trade-off, doesn't it? Especially when you want to deliver bigger things than small drug molecules.
Right. Which takes us neatly into our second main critique point. This one is about integrating solutions for the macromolecular exclusion challenge right into the context of the biomedical applications.
Okay. So the document rightly points out a key strength of aerogels, that tight network of mesopores, usually 2 to 50 nanometers, right?
Yeah, and that structure is fantastic for stabilizing small molecules, amorphous drugs, even nanocrystals. Really locks them in.
But, and the document does acknowledge this, that very strength becomes a major weakness if you're trying to deliver something bulky.
Like therapeutic proteins, growth factors for tissue engineering, things like VEGF or PDGF.
Or even larger gene therapy vectors, perhaps. They just can't easily get into that tight inner matrix. Size exclusion.
The conflict is stated: tight pores good for small drugs, bad for big ones. But the path forward, the solution within the application sections, feels a bit underdeveloped.
It needs a more proactive design focus, I think. So the suggestion here is to reframe it. Don't just state the limitation. Analyze how advanced aerogel design tricks, maybe hierarchical porosity, or specific surface functionalization,
How those could be explicitly used to push aerogels beyond small molecules and into these really high-impact areas like tissue regeneration or complex biologics.
we need to show concrete ways to handle these big therapeutic payloads. You mentioned growth factors for bone repair, RHBMPs2, PDGFBB, things where you often need high loading or specific release profiles.
So how could we engineer the structure? The text mentions cellulose derivatives, BOSC, CNF, things highlighted as good for tissue scaffolds.
Okay, so focus there. Discuss how you could modify those cellulose materials right at the sol gel stage.
To make room for the big molecules?
Exactly. Suggest introducing a deliberate hierarchy of pores. Intentionally build in some larging macropores alongside the existing mesopores, big enough to let those growth factors get in and then get out effectively.
So you're designing the scaffold not just for structure, but for multi-scale transport.
Right. It shows a deeper level of design intent. You're engineering the architecture to manage different size cargo.
And what about chemistry, beyond just making bigger holes? The text mentioned liquid phase modification, adding amino groups to tweak adsorption for ketoprofen, a small drug.
That's the hook. Propose adapting that same idea, chemical functionalization, but target it differently. What if you engineer specific high-affinity binding sites right onto the walls of those mesopores?
Binding sites that specifically attract large hydrophilic protein molecules?
Exactly. You overcome the size problem not just by making the pore physically bigger, but by making the existing pore wall sticky in just the right way for your target protein. Chemical affinity pulls it in, holds it, maybe influences its release.
You turn the pore surface into an active loading zone. That's quite clever. It uses the existing structure but adds a chemical dimension to overcome the physical constraint.
It's a dynamic approach, yeah. Managing structure and chemistry together.
But, and this is always the question with these advanced modifications, how feasible is this when you think about actually making these things? Hierarchical pores, specific chemical sites, does that complicate manufacturing significantly?
Well, that's the perfect lead-in, isn't it? To the final critique, the whole industrial translation piece, the scale-up.
Yes, moving from the lab bench to something commercially viable. Our final point focuses on addressing the industrial and economic feasibility when transitioning from pilot to commercial scale.
And to be fair, the material does touch on scale-up. It highlights a really positive aspect, the environmental benefit. How scaling from, 0.25 liters up to 2 liters cut the environmental impact by up to 96%?
Which is a huge selling point, especially for biomaterials from polysaccharides. Very important.
Absolutely. But the weakness we saw is that the discussion on scale-up seems heavily weighted towards the environmental wins and achieving predictable particle size distribution, the PSD.
And it kind of bypasses some of the harder economic questions and the process engineering logistics, the things that really determine if it can be made affordably and reliably at scale.
Exactly. Like, sterilization is mentioned. It's a necessary final step, right? Often uses ethylene oxide gas.
But the text itself notes that EO gas can be incompatible with sensitive therapeutics, proteins, for example.
Which immediately flags a potential major hurdle, a bottleneck for getting this into a real GMP, good manufacturing practice environment. If your sterilization method wrecks your expensive biologic drug, that's a non-starter.
Right. So the scale-up discussion needs a bit of a pivot. It should definitely include these economic and process engineering challenges. How do you actually solve these issues to get from a successful pilot run to a commercial product, especially minimizing complex, risky batch steps and ensuring everything is GMP compliant?
We need to face the financial realities head-on.
So let's take that sterilization point. Instead of just saying ethylene oxide might be a problem,
Right.
The discussion could suggest actually investigating the feasibility, both technical and financial, of integrating a different sterilization method, something single step.
Like using sterile supercritical fluid processing right within the production cycle itself?
Potentially, yes. That could solve the stability issue for sensitive drugs and maybe streamline the whole workflow. Fewer handling steps, less risk. Now, supercritical fluids, they're not cheap.
True, the equipment and operational costs are high.
But if doing it that way lets you combine, say, drying and sterilization into one clean, contained, GMP-friendly step, maybe replacing several trickier batch processes, well, then the economic calculation starts to look different. It might actually be cost-effective in the long run.
That's a much more robust discussion of scale-up challenges and solutions.
And another good example is coating. Coating is often critical for controlled release. The text mentions using Eudragel, applied in a spouted bed.
But we know water can destroy these delicate aerogel structures, so you need special techniques.
Right, like using compressed CO2 to precipitate the polymer coating, avoiding liquid water contact. Again, often involving supercritical or near-critical fluids.
So the material needs to tackle the economics of that. How can the cost and efficiency of those specialized solvent exchange and coating steps be optimized, simplified for mass production, especially given the known high costs of supercritical fluid tech?
It needs to show a plausible engineering pathway towards making these advanced, necessary steps affordable at scale. That level of practical, market-aware thinking is what bridges the gap between fascinating research and a real industrial process.
Okay, this has been really productive. A deep dive into the aerogel synthesis, the polysaccharide angle, and those biomedical applications.
Yeah, and I think we've landed on 3 key areas for refinement. First, really tightening that link between synthesis control and functional performance, making the why of the process variables crystal clear in terms of clinical benefit.
Second, moving beyond just noting the macromolecule delivery challenge to proactively engineering solutions. Thinking about hierarchical pores or specific chemical functionalization to grab those bigger molecules.
And third, broadening that scale-up discussion. Absolutely, keep the environmental wins. They're important, but also tackle the tough economic and process integration hurdles head-on. Think GMP, think cost of goods.
Excellent summary. So the invitation is to take these points, refine those sections, and feel free to submit the revised material back to us. We'd be very interested to see how these engineering solutions are developed further.
Definitely. Looking forward to it.